Hallo, ik ben Isa de Blauwe, medisch entomoloog aan het Instituut voor Tropische Geneeskunde. In deze video laten we zien hoe je larven van exotische aidesteekmuggen kunt bemonsteren. In onze streken zijn broedplaatsen voornamelijk kunstmatige containers waarin waterkanten stilstaan, zoals banden, regenwatertonnen, emmers, bloempotten, rioolputjes en ondergrondse waterreservoirs. Natuurlijke vijvers, poelen, overstromingsgebieden zijn geen broedplaatsen voor deze exotische muggen.
Inspect the surroundings for stagnant water, preferably in the shadow. Make sure to inspect different types of potential breeding sites. You can find plastic sheets or plastic containers such as kids' toys, plant pots, buckets, rainwater barrels. Also metal containers such as animal feed dishes, a wheelbarrow, old tires. Also inspect below surface, in road drains and catch basins. Give to each potential breeding site type a unique potential breeding site code. Take for each potential breeding site the coordinates and a picture. Back in the office, an overview map is made. Make sure to keep this up to date. The material to perform larval sampling includes scoop nets, seams facing outwards, a flashlight, white balls, pipettes, sampling jars with screw cap, water jerrycan of 5 liters or 10 liters, paper tissues, paper, pencil, scissors and a permanent marker. First, check your container for the presence of larvae. Try not to disturb the water surface and make sure to avoid sudden shadow on the water surface. A flashlight can be used to locate larvae in case the surface is not lightened. The sampling technique depends on the size and type of the container. Small containers such as animal feed dishes, flower pots, kids' toys can be picked up and poured into the white bowl. In case the container is not removable and not accessible with a scoop net, capture the larvae with a pipette directly from the breeding site. Medium-sized containers such as buckets, wheelbarrows, medium tires, road drains are best sampled with a swirling motion, starting at the water surface, continuing downwards along all edges of the container. Don't scratch the bottom to prevent high amounts of debris in the net. Repeat this at least 3 times, but wait a few minutes between each sample to let the larvae come to the surface again. In case of road drains, wait a few minutes after the opening of the cover before sampling to allow the resurfacing of larvae disturbed by the operation. Large containers such as rainwater barrels and large buckets are also sampled with a swirling motion along the side of the container. Repeat this at least 3 times at different locations along the side, with a few minutes break in between. The content of the net is emptied into a white bowl filled with clear water from the breeding site or from a jerrycan, in case of a dirty breeding site. Take out as much of dirt as possible by hand. Leave the white bowl for a few minutes to allow the resurfacing of larvae. Take a pipette to collect the larvae into a sample jar. If possible, take them all. If there are too many, take enough larvae from different sizes and shapes, especially those with a short siphon. Use a pipette with a cut end to collect the large larvae. If there's a lot of dirt in a bowl, you can pour out very slowly some water in another bowl. Make sure no larva leaves the first bowl. This makes the remaining larvae at the bottom more visible. Afterwards, pour the water from the white bowls onto the grass or concrete without leaving standing water. Clean the scoop net thoroughly before you use it again for another type of potential breeding site. Do this by gently knocking the handle against a hard surface. Also clean the white bowl with some clean water and paper tissue. After collecting the larvae, write down the unique sampling code and the breeding site type on the cap of the sampling jar with a permanent marker. Put a small piece of paper with the same unique sampling code, written with pencil, in the sampling jar. After sampling all potential breeding sites, put the clean scoop nets in a bag. Clean the white balls with ethanol. Make sure the sample jars stay upright in a cool box during transport. In case the larvae cannot be transported to the lab immediately, store them in the fridge.